a lot of you will know this anyway, but viruses are one of the most difficult challenges for medicinal chemists. They are super simple organisms. They're not really alive, okay? A virus is not a living thing. It can only reproduce if it has a host cell. But it does have all the information it requires to reproduce itself. But it doesn't have the machinery to reproduce itself, right? So it has its genetic material inside, it has a shell on the outside, it has some glycoprotein spikes, and that's about it. And the DNA codes for the proteins that once it gets inside a cell, it will use to reproduce itself and build its shell. And then it will make copies of itself and go and infect other cells, and so on and so forth. So they carry all the genetic material they need, they just don't have the machinery to reproduce. And viruses are associated with some of the trickiest diseases to treat. A whole range of things from measles and rabies through to herpes and smallpox and HIV. And one thing you'll realize as you look through that list is there are quite a lot of viruses that are vaccinated against. And the first line treatment for viruses, the preferable treatment, is prevention. It's to use your own immune system and vaccinate against them. And it says that at the bottom of page 20, actually, that Pasteur demonstrated the possibility of developing vaccines using an inactivated or weakened form of the virus to prompt your immune system. So that is, and you will have had, many of you will have had vaccines against lots of those viruses, particularly the most dangerous ones. But there are some viruses, HIV would be one example, against which vaccines have not yet been developed. And the reason for that is that they very rapidly mutate their surfaces. They change their shapes and the chemistry is going on those glycoprotein spikes. And that's how your immune system works. It recognizes the surface of the virus. So any virus that mutates its surface rapidly can evade your immune system, even if it's been prompted with an immunogenic agent. And in those cases, we're going to need to think about how medicinal chemistry can try and stop viruses from doing their thing. The problem is, if you want to treat a virus, it's not a living organism. It's not like a bacterium. So with a bacterium, stop it making its cell wall and the thing just dies, right? The virus doesn't live. So you somehow have to stop it from going through its reproduction cycle, and that's something it's going to do in your cells with your machinery. So you've got to try and stop it from using your machinery without stopping your machinery from doing what it really wants to do. And next time we get together, we'll explore some of the kind of rational chemical ways that people go about trying to stop viruses hijacking your machinery. And we'll particularly focus on herpes and HIV. So it'll be like a day of STDs.